So welcome everyone to our seventh, respiratory pathogens webinar series. Today we'll be discussing the Picornaviridae family and specifically Enteroviruses and the Viral Pathogen Resource Picornaviridae page and what you can do with our data and resources. My name is Anna Maria Niewiadomska, and I'm the bioinformatics. I'm a bioinformatics analyst and the outreach coordinator for the Influenza Research Database and Virus Pathogen Resource Database. And joining me today are my colleagues Yun Zhang, a bioinformatics analyst who's done a lot of work on enteroviruses, and Richard Sherman, the director of informatics here at JCVI. And of course, our guest lecturer, Dr. Ken Tyler, who has also done a lot of work on enteroviruses and will be discussing Enterovirus D-68 associated acute flaccid myelitis and new polio, which is the title of his talk. So just to give you a quick idea of the structure, I'm going to give you a brief introduction to the Picornaviridae family, just a basic overview of the virology. After that, we'll have a little time for questions before I move on to a live demo and a tour of the Viper website. And of course, we can stop at any time for questions. And then it's about 9 o'clock Pacific time. Dr. Tyler will start his talk, and then we can have a little bit more time for questions at the end. And this webinar will be recorded, so if you miss it or if you'd like to share it afterwards, it will be on our YouTube channel afterwards. So just quickly before we get started, just to let you know who we are part of the Bioinformatics Resource Centers for Infectious Disease, which is an NIH-funded program that was established in 2004. And the goal of these resource centers is to provide free and public access to not just the data, but also computational platforms and analysis tools for these various infectious diseases, and in particular, infectious diseases that are focused on category A through C human diseases. Which typically tend to be emerging or re-emerging infectious diseases in humans. And as part of our mandate, we perform outreach, and we like to provide bioinformatics training for different tools that we use on the BRC sites, and we go to different conferences. And if you or your institution also has any specific training requests, we would be happy to provide customized training for your group. So don't hesitate to get in touch with us. So right now, when we first started out, there were multiple different BRC groups, but right now we're divided into two main BRC groups. The Bacterial and Viral Bioinformatics Resource Center, which we are part of, and that includes the Influenza Research Database, the Virus Pathogen Resource, which I'll be giving you a tour of today, and our sister site, Patrick, the Pathosystems Resource Integration Center, which in 2 weeks will be giving you a tour of their site and discussing bacterial respiratory pathogens. VUPathDB, on the other hand, tends to focus on eukaryotic pathogens and parasitic pathogens. And we've also had a couple of talks from our colleagues at FungiDB to discuss a couple of different respiratory fungi. So getting started now on Picornaviridae. So this is quite a large family, and it's one of my favorites. It's one of the first viruses that I ever worked on, which kind of got me hooked into virology. But there's quite a lot here, and if you look at where they are part of Riboviria. They're part of the RNA virus families, and they're part of the order of Picornaviralis. And if you look at this tree, you'll see that they're not just in humans and mammals, but there are actually quite a lot of related viruses in insects and amphibians and even plants. So it's quite a diverse and large family. They're pretty small viruses, and they're non-enveloped. They're icosahedral, and they tend to be about 30 to 32 nanometers in diameter, depending on the virus. They're positive sense, single-stranded RNA viruses, and they're non-segmented, unlike influenza. Again, they're quite short. They're only about 6.7 to about 10 kilobase pairs per genome. And again, this varies depending on the family, and they're quite diverse. And within this genome, it encodes a large polyprotein, which then is cleaved into multiple proteins and smaller peptides that form both the structural and non-structural proteins for these viruses. So, as I mentioned, it's quite a large family. There's 63 genera that are known so far, and probably a lot more that we don't know yet. There's 147 species, many of which are still awaiting classification. I've highlighted the ones that are specifically that we know the most about, basically. And the ones in red are those that infect humans, and I'll be discussing those a little bit more. And the ones in black are those that typically infect mammals, but occasionally have been found in human species. So aptaviruses are not as they're rarely have been found to infect humans, but they are well known because they contain foot and mouth disease virus, which infects animals of agricultural importance like sheep and cattle and goats. And so there's been quite a lot of work that's been done on this one. Cardioviruses and Cosovirus infect humans. Enteroviruses we're all familiar with in one way or another because famously they contain poliovirus and other human enteroviruses that we'll be discussing in more detail. Herboviruses mainly infect horses but have been isolated from humans. Hepatoviruses, which contain hepatitis A virus, Cosoviruses, and human pericoviruses. And salliviruses are also thought to infect primates and occasionally have been found in humans, but again, it's not common. So I thought I would just give you a quick idea of what these different genera include. And so, as I mentioned, Aptavirus has 4 different species, and one of them is the causative agent of foot and mouth disease, and it also has bovine rhinitis virus. So, although these tend to not be human viruses, they're very well studied, and there's quite a lot known about them because outbreaks can have huge economic impact. And the clinical manifestations of this are, as the name implies, vesicular lesions in the mouth and feet. They do infect the upper respiratory tract and primarily in split-hoofed animals. And so far, they've been isolated from over 70 different types of species. They rarely infect humans, although young children seem to be particularly susceptible to that. There are vaccines that do exist, but unfortunately, because there are so many different species and serotypes, it's hard to have cross protection. And immunity seems to be not long lasting. So next we have the cardiovirus genus. There are 6 species of cardioviruses, A through F. Including Thiele's murine encephalomyelitis virus. There are multiple hosts for these viruses, and I believe Cardiovirus B is the one that infects humans. And in general, like a lot of these different Picornaviridae, they either can cause mild symptoms in most, such as gastroenteritis or flu-like symptoms, but occasionally they can cause or are thought to cause non-polio-associated acute flaccid paralysis. And Saffold virus is one of the more recently identified ones in children, and this was found in association with gastroenteritis or upper respiratory tract infections. And again, sometimes the correlations are there, but it's hard to make the direct link between the virus and the disease. Cosovirus is another recently identified picornavirus. There are 5 species that are known so far, A, B, D, E, and F. And the pathogenesis in humans is unclear. They've been isolated from patients with gastroenteritis, and it's suspected that they are the cause. And they are thought that there is a hypothesis that they may cause these viruses may cause more severe disease in immunocompromised patients. So hepatovirus disease, this is also known as the causative agent for hepatitis A, which you can catch just from, if you haven't been vaccinated, from just contaminated food or water. And there are 9 species recognized, and they've been isolated from multiple different mammals. But this is definitely one that can cause disease in humans as well. Although symptoms are typically mild, it can cause acute liver failure in older people as well. However, there is an effective vaccine for it, although no treatment exists. So Pericoviruses are another genus of Picornaviridae that have been isolated from humans, as well as from ferrets and rodents. There are 6 species, A through F, and Pericovirus B is the one that's thought to infect, A and B are thought to infect humans. And again, like many of these picornavirus genera, they can cause mild symptoms, either in the gastrointestinal tract, or they can cause respiratory symptoms. And rarely, some of them can cause sepsis, meningitis, or other neurological symptoms like encephalitis. And again, there's no specific vaccines or treatment for these. So Cobovirus and Sallivirus are 2 other genera that have been isolated from humans as well as other mammals. And again, they cause mild gastroenteritis. And there's not much that's known about these, so I'll just leave it at that. So enteroviruses are the most well-known and best characterized genus of, I would say, picornaviruses. It's quite a large group. There's 15 species, and these include enteroviruses A through L, as well as rhinovirus groups A through C. And member species include Coxsackie-viruses, which are the causative agents of human hand, foot, and mouth disease, enteroviruses, one of which, of course, is poliovirus, echoviruses, and human rhinoviruses, which cause upper respiratory tract infections, and most of us have probably experienced. So in humans, it's thought that rhinovirus A and B and C cause infection in humans, and for the enteroviruses, it's thought that groups A, B, C, and D are the ones that circulate in us the most, and the other groups, E through L, tend to be isolated from other mammals or other species for the most part. So as I mentioned, Coxsackie virus A16 is the main causative agent of hand, foot, and mouth disease, although there are other species as well. Enterovirus C includes the poliovirus 1 through 3, as well as many other Coxsackie viruses. And Enterovirus D-68, which belongs to the group D enteroviruses, usually causes mild to severe respiratory illness, but it has been associated with acute flaccid paralysis or myelitis, which we'll be discussing later on in this talk as well. So in general, most of these enterovirus species cause mild cold-like symptoms. They last about a week. More serious symptoms, however, can include hypoxia, meningitis, conjunctivitis, hand, foot, and mouth disease, and paralysis. So while they generally are thought of as mild childhood infections, there can be severe consequences in a small percentage of patients, which is why we spend a lot of time and effort trying to understand these viruses, study them, and develop vaccines for them. So Picornaviridae tend to have very similar variant structures. They're non-enveloped, unlike most of the other viruses that we've discussed in this series. And they tend to have a very geometric structure. They're spherical, and they're packed together usually with 4 structural proteins, VP1, VP2, and VP3, which are on the outside and form this triangular pattern. And together, they form 60 protomers, each consisting of 4 polypeptides. VP4, which is on the inside, is the 4th one, and it's located on the inside part of the capsid and just holds everything together. So this is the genome structure of Picornaviridae. Again, it tends to be a very short, compact virus, but it packs a lot in there. It forms 1 polypeptide, which goes from pretty much the beginning of the genome till the end, with the exception of the 5' and 3' untranslated regions. And this is divided into about 3 different regions. P1, which contains the structural proteins, P2, and P3, which contain the non-structural proteins that are associated with replication and modulation of host response. So after this polyprotein is translated, it then gets cleaved into a series of smaller proteins, and these in turn are cleaved into more proteins. So one thing that I should mention is that there's an additional protein L over here, which is the N-terminal leader protein, and this is only present in some genera. And it either has a papain-like cysteine protease function or another function. But for the most part, these Picornaviridae have similar structures. So one thing I didn't mention is that unique to, or one of the special things in Picornaviridae is that they contain this protein, VPG, which is translated from the P3 region. And together, along with this RNA structure in the 5' UTR region, they initiate translation of the polyprotein because it acts as a 5' cap. So rather than engaging in cap snatching or using host mRNA as a 5' cap, it uses the IRES region, the internal ribosomal entry site, as well as the VPG protein as the primer, as the initiation factor to bring together all of the translation initiation factors that assemble either directly on the first start site for the polyprotein or upstream of it and then go on to scan the viral RNA for the first methionine coding region. So after that, once the translation nucleoprotein finds uh, the start site on the viral genome. This goes on, translates the entire polyprotein until it finds the poly A tail at the 3' end that initiates termination. So one thing I just wanted to mention before we move on is that while these different Picornaviridae genera have a similar structure, of course, if you're looking at your own virus of interest, it's going to differ slightly. And if you look here in this picture on the right that I've pulled from this publication, it'll give you an idea of the various differences between them. So while they might have the same structural proteins that are conserved, if you look in the P2 and P3 region, you can see that there's different cleavage points, different cleavage sites, and there's occasionally alternative cleavage. And of course, you have the leader protein that I mentioned in some genera, such as Cardio, Apto, Erbo, Kobu, Tesco, and Sapello and Seneca virus. So the entire polyprotein is translated, and then there are viral proteases that will cleave the structural proteins, the P1, into the various proteins, VP1 through 4, and P2 region is usually cleaved into 3 different proteins, although there can be differences depending on the genera, as you can see here. The precursor 3 polyprotein region is usually cleaved into 4 proteins, which also includes the VPG protein, which then moves on to act as the translation primer for the virus. And some enteroviruses, I should also note, have an upstream open reading frame as well. So moving on to the viral replication cycle, it's a little bit different than enveloped viruses in that attachment happens directly onto the viral capsid, and this mediates endocytosis. After that, capsid conformational changes induce uncoating of the virus, and the RNA then moves into the cytoplasm. The VPG and the IRS initiate translation. The polyprotein is processed into multiple different proteins. And one of the functions of these proteins 2A is to shut off host mRNA translation so that only viral mRNA becomes translated. Eventually, these proteins facilitate genome replication of the virus, and together, along with the structural proteins which form pentamers and provirions that package the new viral genomes, these are then released either through lytic or non-lytic processes. Obviously, the lytic processes destroy the cells, and that's one of the reasons why you end up with paralytic diseases. So where does enterovirus genomic diversity come from? So first of all, there is a high level of genetic diversity even within a species. So subspecies are usually classified based on the major hypervariable capsid protein in VP1. And even in enteroviruses, there are 326 different types that are scattered across the 15 species. And this includes 168 different rhinovirus types, 3 poliovirus types, and 103 enterovirus types. Picornaviridae are also known to experience both intergenotype and intragenotype recombination. And an example of this can be seen in one of our colleagues' publications and our publications. And these are known recombinants, where you can see that there's obviously a part of the genome that's been acquired from another enterovirus. And there are also several new recombinants that we were identified. And you can read more about this in this paper. So it's a little bit hard to try to summarize enterovirus genomic diversity in a short talk. So again, I'll direct you to a paper my colleagues and our collaborators wrote on the epidemiology and evolutionary diversity of non-polio enteroviruses, and they do quite a thorough job of it. So we know that Enterovirus A is responsible for most hand, foot, and mouth diseases. Hand, foot, and mouth disease cases, Enterovirus B. There are 59 different types, and they're mainly responsible for acute flaccid paralysis cases. Enterovirus C is generally uncommon, and that's this group here. And they can be the causative agents for things like conjunctivitis outbreaks. And of course, Enterovirus D up here, which recent outbreaks have been responsible for acute flaccid paralysis as well. So again, enteroviruses are quite a large group of viruses. They're very diverse, but they're not just diverse in their genomic composition. They have quite a diverse distribution in terms of both their geographic spread as well as their temporal spread. So you can have different enteroviruses circulating in different parts of the world, and you can also have different outbreaks of enteroviruses in different years. And again, that's summarized in a really nice publication, which I referred to previously, on the epidemiology and spatiotemporal distribution of non-polio enteroviruses. So you can see, for example, in North America, most of the sequences were Enterovirus D-68 between 2000 and 2018, whereas in Asia, most of the circulating enteroviruses belong to different Coxsackie species, or Enterovirus 71, A71. And similarly, if you look at the temporal distribution, you'll see that different enteroviruses are responsible for different outbreaks throughout the past couple of decades, where you'll see species, the number of sequences in our database spiking depending on what outbreak is currently circulating. So for example, in 2010, there was an enterovirus A71 outbreak responsible for hand, foot, and mouth disease, or Coxsackie virus A6 a few years later. And similarly, you'll see similar patterns for other enteroviruses, Enterovirus B, C, and D, which are in this graph down here. So it's a lot to take in, but it's worth taking the time to read about it because it's just such a diverse and interesting group of viruses. So what are our options in terms of therapeutics? Right now, there are no antivirals that are approved by the FDA for use against enteroviruses or picornaviruses in general. Pleconaril, which binds the VP1 protein and prevents uncoating or attachment, has had limited clinical use, but due to toxicity issues, was never approved. There are a couple of other types of antivirals and viral inhibitors that have been used, including capsid inhibitors, the 2A protein inhibitors, a lot of protease and polymerase inhibitors. There's a couple of nucleoside analogs that have been used as well. So there's There are antivirals that have been explored for every single step of the viral life cycle, but up until now, none of them have been approved or reached end-stage clinical trials. So, um, yeah, there's a little bit of experimental evidence that ribavirin, which is a common antiviral, might be effective, but so far the evidence is only has only been shown in mice. But one of the things that you can use the Viper site for is to look at the crystal structures that have been generated for these different antivirals and the structural proteins of these Picornaviridae. So, for example, over here, you can see the structure of human rhinovirus 16, which is in complex with pleconaril, which is this antiviral drug that was in clinical trials at the time. And in green over here are the ligand binding sites for pleconaril. So what vaccines are available? So we've mentioned a couple of these, and these are obviously a little bit more positive. So there are vaccines for foot and mouth disease, but again, these are not cross-protective against all of the different types. Candidates exist for some cardiovirus vaccine, for some cardiovirus species, but again, these haven't been approved, and this is for porcine encephalomyocarditis virus in pigs. There are none for Cosovirus and none for Pericovirus. There are quite a few that either have been developed or in development for Enteroviruses. Again, famously, polio vaccine is well known, and most of us have hopefully had it. And there's more recently a vaccine for Enterovirus 71, which causes hand, foot, and mouth disease. And this was put into market in China, and I think there's a couple others as well that were released in China, but so far these haven't been approved in the rest of the world, but they so far appear to be effective against severe hand, foot, and mouth disease in children there. But they're also developing a double vaccine against both Enterovirus A71 as well as Coxsackie-virus A17, which also is a causative agent for hand, foot, and mouth disease. And they're trying to develop combination vaccines for other species so that it's cross-protective. There are several candidates in development for Enterovirus D-68. There have been multiple trials and candidates for rhinoviruses, but as far as I know, none have been approved or exist yet. And finally, there is a vaccine for Hepatitis A virus, or several vaccines for Hepatitis A virus. So that wraps up my whirlwind tour of Picornaviridae. I'll just remind you that we have one last webinar in our series on bacterial respiratory pathogens and the Patrick site that will be given by Dr. Rebecca Waugham in a couple of weeks on the 1st of June. And again, if you've missed any of our previous talks on other viruses such as influenza, coronavirus, and RSV, as well as our partners talks on coccidioides and Aspergillus and AMR, you can find those on the various BRC YouTube channels. And if you can't find them, please email me and let me know and I'll direct you. And then just finally, one last thing I wanted to let you know about is that NIAID is sharing a survey on the usefulness of the Bioinformatics Resource Centers. So if you do use any of our sites, we would really value your opinion and would appreciate you letting us know how we can improve what we do for you. And that is it. I will take a break and check for questions. Oh, and if you can't find if you don't have time to copy this link, you can go to the Viper website and you can find it, the survey right at the top of the virus news section. So you can find the Virus Pathogen Resource by going to viperbrc.org. And this will take you to our portal page where you can explore the different viruses that you might be interested in looking at. And if you scroll down, you'll see our virus families divided by the virus types. So single-stranded positive sense RNA is what we're interested in today. And you can go ahead and click on Picornaviridae if you'd like to explore this family. So the basic structure for the Viper site, if you're not already familiar with it, is a section where you can search for different types of bioinformatic data, an analysis section where you can explore the different tools that are available for bioinformatic analysis, and a workbench where you can save your data, share your data, upload your own working sets, and combine them with Viper data and generate your analysis there. You can also share the results of your analysis with collaborators and anyone else who might have an account. So you don't need to have an account to use the website, but if you do want to share your data for more than, if you do want to save your data for more than 24 hours, you should register so that you can save it there. And of course, the same goes for uploading data. Of course, everything that you upload is private, and we do not make any of it public. So I'm going to go ahead and click on search and just give you a quick idea of the different types of data that are available. The most commonly used search feature is the sequences and strains. So whether you're interested in rhinoviruses or enteroviruses, or you happen to work on foot and mouth disease, all of the different genera are available here, and you can find them by scrolling through the taxonomy tree. So the different viral genera that we discussed are here, and you can expand and collapse them as needed. So if I was interested, for example, in enteroviruses, I could either select them all or select the subspecies that, or the species that I was interested in. Let's say Enterovirus D. I could go ahead and click on select all. And then,
Anna, sorry to interrupt.
sure,
Could you maybe zoom in a little bit? It's a little harder to see.
sure, no problem. How's that? Okay.
Yeah, maybe one more. Yeah, perfect. Thank you.
Okay. So we return the number of results that you would get depending on your selection in real time, so you can get an idea before you see your search results table. You can search by collection year, and one of the things that we do at Viper is that we standardize the metadata. So whether the dates were entered, let's say in a US format versus European format, they're all standardized. We standardize geographic regions and countries so that it's all easily searchable. And we standardize also the host metadata so that, let's say, for example, a virus was uploaded as swine and one was uploaded as pig and one was uploaded as porcine. You would be able to find all of those underneath the common name of pig, for example. And so we curate those and standardize them so that it's all easy to find. And you don't have to deal with parsing through the scientific name and the common name. So let's say, for example, I'm interested in enteroviruses from 2014, the 2014 outbreak in humans. And let's say I want those in North America and specifically those that were isolated from the United States. So that would return 935 sequences. And one of the nice things about the Viper sites is that you can also specify that you want complete viruses only. So oftentimes people will upload fragments, which might not be what you're looking for, especially if you're looking to do a complete genome alignment. So if you specify complete genomes only, this will bring our results down to 238 only. We do have a couple of extra features, particularly in regards to rhinoviruses. One of our collaborators had a surveillance program where they would collect a lot of extra data on rhinoviruses. So here you have human rhinovirus A, B, and C. And there's some extra metadata, including the clinical diagnosis and association with clinical features such as asthma or cold test scores. And it also includes host age and host gender. There's a few other things in the advanced options that I would encourage you to click through, and that's, for example, being able to specify the month range that you're interested in, or searching through specific genomes and strain names that you're interested in. And one of my favorites is removing duplicate genome sequences, which, especially if you have a large data set, can be quite handy and help shrink that for you if you're not interested in duplicate sequences. So if you go ahead and click on search, that will provide you with a results table where you can view the different columns, including the strain name, the virus type. It'll give you the GenBank accession number, your sequence length, the collection date, and all the other host metadata. And you can sort by these headers as well, just by clicking on them. And you can also customize the display settings just by clicking on display settings and add and remove the different columns that you might be interested in. And if you just go ahead and click on view one of these, it will take you to a page that gives you all the details on that specific strain, including the strain information and metadata, the information pulled from GenBank as well, and give you a quick map of the genome and the different proteins that are there. You also have all the protein information that's provided. Let's say you're only interested in the VP1 protein. You can go ahead and click on that, and that should give you the amino acid sequence, but the different domains that can be found in the protein as well. So going back to our search page, if you're interested in immune epitopes, you can go ahead and click on that. And this will give you a list of immune epitopes for the specific viruses that you're interested in. And these can either be experimentally determined epitopes, which have been proven, and these are derived from the immune epitope database, or we have predicted immune epitopes that are predicted by an algorithm called NetCTL. So going back to the experimentally determined epitopes, these can either be B cell or T cell epitopes, and you can further specify what you're interested in by clicking on a couple of the other options here. And again, if you click on advanced options, you can specify the MHC allele class that you're interested in, or the specific amino acid range on a certain protein that you might be interested in looking for immune epitopes in there. Okay, so going back to the search, I'm just going to show you one more thing here. If you're interested in looking at the protein structures for a specific Picornavirus genus, you can go ahead and click on 3D protein structure search, and these are all the 3D protein structural files whether they're X-ray crystallography or NMR that have been imported from the protein data bank. And let's say you're interested in enteroviruses, you can open that up, select everything that you might want to see for Enterovirus A. And so we have about 78 structures over there, and click on search. This will return a list of the different structures that are available. So for example, there would be the structure for an entire capsid or a specific protein, and the table will tell you how they were generated, whether it was through electron microscopy or X-ray diffraction, which is X-ray crystallography. And there's also links that will take you to the protein accession number. So if you're interested in exploring these, you can just click on View the Structure. And there's a built-in structure viewer on the Viper site. And you can change the options. My favorite is looking at it in cartoon structure. And you can just turn it around directly on the site. Click on Spin or Rock to just get an idea of what the protein looks like. If there's ligands, you can, there's an option where you can click on Highlight the ligands to see where they might be. And a couple of other things that you might be interested in. So let's say if you're interested in a specific region of the protein, you might want to color it by, let's say, looking for chains or a specific amino acid residue or structure type. And you can also look at hydrophobicity, for example, or charge. Okay, so we're getting close to 9, so I'll just run you quickly through the different tools that we have available before we move on to questions and our guest lecture. We have some traditional bioinformatic tools that you're probably already familiar with, such as BLAST. You can also look for primers and design your primers using the tools here. You can perform multiple sequence alignments, look for sequence variation. You can generate phylogenetic trees. One of the tools that was particularly useful during D-68, Enterovirus D-68 outbreak was the Metacats tool, which performs comparative analysis based on specific metadata. So I think we use this to compare specific groups of viruses that were causing neurological symptoms versus those that weren't, and identified several residues in those viruses that turned out to be causative in terms of their ability to infect neurons. And you can also use these tools to generate phylogenetic trees, and I think we have some beautiful examples of those in the publications that I referenced. And I also have some examples in my workbench. So, for example, I have just a reference tree over here of some enterovirus sequences. And you can just click on View Tree after you build it, and this will generate a tree in Archaeopteryx, which is built-in software on the Viper site. And this might not look like the most interesting tree at the moment, but if you take the time to explore these, they're extremely customizable. You can change the colors, you can change the groups. You can also color it based on the data that's already input into the Viper database. So it's really handy to be able to do this directly on the Viper site and have the metadata already integrated into the sequences as well. So that is it for me. I think I'll leave some time for questions. And
All right, well, Anna, if you're ready, I'll go ahead and introduce Dr. Tyler. Yeah, it's my real pleasure to introduce Dr. Kenneth Tyler as our guest speaker today. Ken is the chair of neurology at the University of Colorado Medical School. He got his undergraduate degree from Harvard University and his medical degree from Johns Hopkins University School of Medicine, and he did his residency training at Mass General Hospital. Ken is a true physician scientist. He really tries to organize his research program around real-world infectious disease problems. He's really focused his research on virus infections that affect the central nervous system, including a wide range of different viruses, from flaviviruses like West Nile virus and Zika, herpes viruses, and as well as enteroviruses that he'll talk to us today about. I got to know Ken because his group developed a really valuable mouse model of paralysis caused by Enterovirus D-68 that has been really a tremendous tool for us to understand the pathogenesis of enteroviruses, especially as they affect the central nervous system. So Ken, we're really honored to have you here and look forward to your presentation.
Well, thank you, everybody. Thank you, Anna. Anna gave a great setup for most of this and is going to, I think, run the slides, because you'd think after a year I'd be able to make WebEx work, but I guess I'm a Zoom maven, not a WebEx maven. One quick disclosure, we've been working with a company called Zab Bio with Miles Brennan and some of his colleagues to develop some humanized monoclonal antibodies against enterovirus that I'm going to briefly mention. And we've been very grateful for the support that we and people we've collaborated with in the lab have gotten from the NIH for this work. So maybe next slide. I wanted to start just quickly and mention the people, who have really both got me involved in this and done the great work that I'm sort of going to be the voice of. On the top row there, you can see Kevin Mezikar, Dan Pastula, and Terry Schreiner. Kevin is known to many of you in the enterovirus field. I'm the co-mentor on his K23 award, and he's a pediatric infectious disease faculty member here at Colorado, who's done just great studies on the epidemiology of Enterovirus D-68. Dan Pastula worked for the CDC and now still does some contract work for them, but is part of our neuroinfectious disease group and was one of the CDC officers originally investigating AFM, acute flaccid myelitis, when the first outbreaks occurred. And Terry Schreiner is also part of our department. She's a wonderful neuroimmunologist and pediatric neurologist who takes care of many of these kids, and was the one in the summer of 2014 who asked me to walk across our campus to the Children's Hospital to see the first cases of this, which got me involved in what is truly a bedside to bench and back kind of story. And then in the bottom row, there are just three people from the lab that I've highlighted. Allison Hickson is completing this in a month or so, the MD part of her MD PhD. The PhD is already done, and she's going to go on and be a neurology resident at WashU in St. Louis. And she helped develop this mouse model. It was, in fact, her rotation project as an MD PhD student. And then Mike and Josh there have been instrumental in some of the studies with antivirals and antibodies that I'm going to talk about. Next slide. So The story for me actually began in the summer of 2014 when the first case of what would be this recent series, sort of the index case in a sense, of AFM was admitted to the Children's Hospital in Colorado. And it was very clear within a month that something weird was going on, in that we saw a cluster of originally about 9 cases of young children who developed a polio-like illness that I'll tell you more details about in a second. And this led us to notify not only our own State Department of Public Health, but the CDC, led to a quick report and what was called a health advisory notice to see whether this was a local Colorado problem or a more national one. Next slide, Anna. We already had a little bit of a foreshadowing or a clue as to the epidemiology because this occurred in the midst of a surprising and new emergent outbreak of respiratory infections in children that was subsequently shown to be caused by Enterovirus D-68. And if you look just at the line part, not the bar part for a second of that graph, you can see in the sort of green-blue line there that in a typical year, the Children's Hospital at Colorado might admit about 20 patients per week who came to the emergency room for respiratory infections. And if you look at the red line, you can see that in the summer of 2014, this number had increased by anywhere from 2 to 3-fold. There was this deluge of these respiratory cases linked to Enterovirus D-68. And it was in the middle of this, now look at the bars, that these cases of this polio-like pediatric weakness occurred. And so, of course, from the get-go, we all kind of thought, sure, this could be a coincidence, but it was a likely linked event. Next slide. This is from the CDC's website, and I want to give a shout out. I think when I was looking at the list of participants, Janelle Routh was on the call from CDC and Sarah Kidd, and they've just done a spectacular job in pulling together investigators, as Richard alluded to, and in collecting clinical and other data on AFM, and it's just been really fun interacting with them. This is from the CDC's website and just shows the number of cases and when they've occurred of acute flaccid myelitis, which was sort of defined as the acute onset, initially in children, but now there's sort of an open age definition of an acute flaccid weakness associated with a particular type of abnormalities on MRI that I'm going to show you about. And I think all of you can see quickly 2, or maybe we could even call it 3 features of this slide. One is that the outbreaks of AFM seem to occur in an every other year kind of model. I'm sorry that some of the numbers on the top are a little bit frame shifted there, but in 2014, there were 120 documented cases, confirmed cases, quiet in 2015. The next spike was 153 cases in 2016. Quiet year in 2017, a big peak of 238 cases, which should be over the spike in 2018, and then things have been quiet. So it was a predominantly summer and fall seasonal pattern that occurred every other year. And of course, we thought that 2020 would be an even bigger outbreak because each spike had more cases and involved more states. But in fact, it was stunningly quiet, just the sort of basal level of cases. And I can tell you that 2021 so far has also been reasonably quiet. I think there are a total of 16 cases that are being investigated, 5 confirmed, 11 still under investigation. So we actually think that it may be COVID-19, social distancing, mask wearing, and other features that prevented that 2020 spike. And we're all kind of waiting with bated breath to see what 2021 will look like. Next slide. This is just from 2018, and just to quickly make the point that virtually every state in the United States, you can see there are a few that are in the lighter blue, didn't have cases, but almost every state in the US reported cases of AFM. And California was actually one of the bigger totals with 15 in that year. Next slide. I think this one may be a steal from Janelle. She can comment if she's still on the line. But I wanted to make the point that as they started to accumulate both at the CDC level, but also from local hospitals, we had a paper, others did as well. It quickly became clear that almost all of the patients who developed AFM according to the defined epidemiologic criteria had an antecedent febrile illness, usually respiratory, occasionally GI, and a little bit sort of similar to respiratory, just a pure febrile one, that typically preceded by anywhere from 2 to 5 days the onset of the neurologic disease. So this was, frequently referred to as a viral-like prodrome. So again, was another piece of evidence in the epidemiology that this might be a viral syndrome. Next slide. I won't go through the chart there in gory detail, but I just want to tell you that the neurologic part of this, and this is from a review we did in the Annals of Neurology, is the children developed the relatively acute onset of weakness. The limbs were flaccid, by which we meant the tone was decreased. The reflexes were absent. The weakness was often asymmetric, meaning it wasn't equally distributed across the 4 extremities. There was a tendency for the upper extremities to be involved more than the legs, and for the weakness to be proximal rather than distal, meaning more, say, at the shoulders or in the hips than in the hands or the feet. And sensation was typically intact in the overwhelming majority of these children. And so to a neurologist, when you describe something like this, their first thought is, oh, that anatomy represents involvement of the motor neurons in the spinal cord, or less commonly the motor nerve roots, because you get this what's called a lower motor neuron pattern of neurologic findings, and there's no sensory involvement. Next slide. Another clue as to the potentially inflammatory nature of this came from a number of studies. Oops, I don't think I put the reference here, so this may be, again, a CDC source. The spinal fluid, when examined in these patients, very, very frequently showed a pleocytosis. Spinal fluid usually is a pretty bland fluid that has very few, if any, white cells in it. And you can kind of see that the majority of patients, when the spinal fluid was examined, had a pleocytosis. Over 80% did, more than 5 cells, usually lymphocytes. And this is the classic pattern we see with viral infections. So that was another piece in the puzzle, that this was likely infectious or inflammatory, along with the prodrome I mentioned. Next slide. If you looked at MRI imaging of the spinal cord of these patients, what you saw was these dramatic, longitudinally extensive lesions. And if you look at that far left panel with the black arrows there, all of that white being pointed to by the arrows, and I think Anna's helping me out a little bit there with her cursor, is abnormal signal in the spinal cord, which is also swollen. And if you look in the middle section of the slide there with what looks like those owl eyes almost in that bottom panel, those are right in the middle of the anterior horns of the spinal cord. So the pathology matched very well with the clinical features, and again suggested a motor neuron and spinal cord process, occasionally with lesions elsewhere. Next slide. The children affected by this were typically young. The age varied across the different years. I'll show you some pictures in a second. This one stuck in my mind because DH was one of the oldest children. He was about 17, as I recall, when I first saw him, a senior or a junior, as I remember, in high school, and had been a vigorous soccer player and athlete, and developed this tremendous upper extremity weakness with loss of bulk in the muscles, as you can see there, of the around the scapula, the upper shoulders. You can see how prominent his clavicle is, his scapula is. You can see all the atrophy in his chest muscles, and even a little feeding tube that he needed. And one of the other features that was quite striking about this illness was not only how profound the weakness was, but how poor overall the recovery was. So this was actually taken a year after his original infection. And although he'd had some improvement in function, it was relatively modest. So this was indeed a sort of scary thing, to have children paralyzed and to have an illness that they often showed a relatively modest recovery from. Next slide. Just look at the left-hand side of this slide. If you look at panel D there, that was again at about a year, a young boy who was affected by this, and you can see the profound atrophy in his right leg muscles compared to the more normal-looking left ones. And then in the panel C there, again in about a 1-year-ish follow-up, this boy is being asked to raise both of his arms above his shoulders, and you can see he gets his right arm up great, but can not even get his left arm up to 90 degrees. So this was a pretty dramatic illness where at least the Enterovirus D-68 associated cases often had a relatively modest improvement. Next slide. So some of the clues suggest that epidemiologically a link to Enterovirus D-68. Others suggested infectious and inflammatory, as I alluded to. And when you looked at body fluids, notably things like nasopharyngeal or respiratory specimens, and did RT-PCR on them, the virus that was most commonly isolated, especially if one obtained specimens early, was Enterovirus D-68. There were occasional isolations of other viruses, but that was the overwhelming favorite. You can see there, if you just kind of look at the parts of the bars that are colored in blue, those represent the total fraction of Enterovirus D-68 that was found. And in the upper series, it's days from onset of respiratory or febrile illness. And if you just look at the lower graphs, that's days from limb weakness onset to specimen collection. So this provided a sort of partial smoking gun. Enterovirus D-68 was the most commonly isolated virus from patients meeting criteria for acute flaccid myelitis. But you'll notice I'm not showing you spinal fluid. And one of the things that was obvious very early was that unlike these data, it was pretty hard by PCR or by other methods to isolate Enterovirus D-68 genome, etc., from the CSF of cases. Only a few % of patients were positive. And so there was still some query about, wait a minute, could this be coincidental? How come we weren't seeing more virus in spinal fluid the way we would, say, with poliomeningitis or something like that? Next slide. Two papers that came out from two different groups, basically in the summer of 2019, helped us move this needle a little bit further. Mike Wilson and colleagues at UCSF and Ian Lipkin and colleagues at Columbia used different techniques, and I won't go through them in detail, but used different techniques to probe spinal fluid for antibodies against enterovirus in patients with AFM AFM versus controls. And in both cases, they found significantly increased incidence of those antibodies in the patients versus the different control groups. And so this was a little closer to a smoking gun. It's still unfortunate that we have very limited pathology from these cases. Fortunate for them, they don't usually die acutely. A few cases have died many years after or years after onset of infection. And we still haven't had formal proof of intrathecal synthesis, meaning you're making more antibody in CSF than in the periphery. But I think almost all of us in the field believe that Enterovirus D-68 is the primary driver of those pediatric seasonal spikes in AFM that I showed you earlier. Next slide. So shortly after I saw those first cases, Allison Hickson came to the lab, and I hadn't given as much thought as I should have to what her MD PhD rotational project should be. And she said, "So what am I going to do?" And of course, you know, I hadn't thought about it at all, so I had to think fast on my feet. And I had just come back from children seeing these cases. So I made a really stupid suggestion. I said, let's work on a mouse model of AFM. And she said, okay, because she didn't know anything better. And we got a bunch of these isolates and started to stick them into mice. And I'm going to show you in the next few slides some of what we found that Richard already alluded to. This is just some data using one of the 2014 strains isolated from Illinois. And in the colored graph on the top there, I just want you to see the y-axis is a paralysis score. Higher numbers are worse. The x-axis is time. And the more virus you gave to these mice, this happens to be an intramuscular injection in outbred Swiss mice, the quicker they got paralyzed and the more severe it was. We also were able to show that different routes of infection produce different frequencies of paralysis. The lowest rates were if you tried to mimic what I think was natural infection and did things like intranasal inoculations, where you can see on the right-hand column there in the blue background that only about 3% of mice inoculated intranasally developed paralytic disease. But if you gave it IC or IM, intracerebrally or intramuscularly, you could approach 100%. Now, we also know that there were some host factors that were likely important because there was also a hierarchy of mouse susceptibility across different strains. And outbred mice like NIH Swiss were relatively resistant, whereas mice like BALB-C were relatively sensitive. Next slide. There was also a pretty striking age-related susceptibility, mimicking what we were seeing in the human disease. If you open up the definition of AFM to all age groups, you find that somewhere around 10% of cases that meet the syndromic criteria occur in adults, but the overwhelming majority are in young children. And by the way, if you look at those adult cases, they often aren't caused by Enterovirus D-68, but by other viruses, including West Nile or even Enterovirus A71. Next slide. I don't know if this will play through WebEx, but Anna, can you see I had two movies in here of the mice, so I don't know if you, whoops, I guess we won't be able to see, oh well. You'll have to take my word for it. I was going to show you mice. You can see a little bit on the lower guy that he's dragging his hind limbs there. This was at about day 12 after inoculation, and this one up at the front had a paralyzed forelimb. And I apologize that being a Zoom maven but not a WebEx maven, Anna can't show you this because she's downloading this from Dropbox. Next slide. If you took the spinal cords out of those animals, you could find both infectious virus and viral genome that tended to peak about 6 days after infection, as shown here. Next slide. And there was a wonderful correlation between the clinical pattern of paralysis in the mice and the pathology. So here you can see this poor mouse had a paralyzed left hind limb. This was with another one of those 2014 strains from Missouri, and the black arrow was just to remind me it was his left hind limb that splayed out there. If you look on the right-hand side of the slide, you can see on one part of the spinal cord where the letter R is for right, lots of preserved green choline acetyltransferase staining motor neurons. But if you go to the other side of the slide, the motor neurons are gone. In the area corresponding to the innervation of the paralyzed muscle. And if you look closely in the surviving motor neurons and you stain for a Enterovirus D-68 antigen with an antibody against the VP2 protein, you can see those surviving motor neurons contain viral antigen. Next slide, it shows it a little better, I think, Anna. That bright green cell there is stained positive for Enterovirus D-68 and is right in the anterior horn of the spinal cord. Next slide. If you do electron transmission, electron microscopy from spinal cord specimens, you can see that the neurons are jammed packed with enterovirus-like virion particles. And you can see I've shown you 3 different magnifications. And then just from the CDC's database, a little in the lower right-hand corner, a reference specimen of what Enterovirus D-68 virion particles look like. And you can see these totally meet the morphologic appearance of Enterovirus Enterovirions. Wouldn't tell you, of course, which type. Next slide. When we started to do this, we also started to inject into mice many different isolates that we got from different sources, including some from California, some from New York, some locally. And it was clear that many of the contemporary, as we call them, isolates from 2014 could induce paralysis in mice. So tall bars are just percentage of mice with weakness. But there are 2 things I also want you to notice. One is there was something different because when we looked back to the original prototype strains isolated in the 1960s, Furman and Rinn, we got almost no paralysis when injecting those into mice. So there did seem to be something different about the modern versus the so-called ancestral strains. And then you'll notice there's almost like a little hole in the bar graph there where California 144231 is. And I want to make the point that not all of the modern isolates, when injected into mice, produced paralysis. Now, originally, there was some thought that maybe specific clades, and Anna showed you some of the more modern cladotypes or phylogenetic trees. It's clear now that at least in the mouse models that viruses belonging to essentially every clade and subclade can produce paralysis. Different clades and subclades show up more frequently in the human material, likely not so much reflecting their paralytigenic potential, but more what virus is dominant or circulating at a particular time and in a particular area. Next slide. We've started, and this is preliminary work, so could change. Whoops, looks like it disappeared for me. I was going to say we've I'll have to get Anna to come back. There we go. We've started to generate chimeric viruses. What I want you to see here is in red, you can see here's one of those very paralytic strains, Illinois 52. And I showed you that hole in the bar graph before with California 31, as we'll call it for shorthand, which doesn't induce paralysis. Those two strains only differ from each other by 11 amino acids, of which 5 are in the capsid proteins, VP1 and VP3 shown with the black arrows, and an additional 6 are in other non-coding proteins. There are also a total of about, I think it's 7, I can't count dots so easily without my glasses here, 7 changes in the 5' UTR region. We've started to make and test some of the chimeras between those two viruses. And I think you can see here that if you take a virus that has the whole right-hand portion of the virulent IL-52, but all the capsid proteins plus the UTR plus 2A and 2B of the attenuated California virus, it remains attenuated. If you swap out the UTRs, you do get a little bit of a virulent signal. You can see there that instead of 0% paralysis, you maybe get 13. And conversely, if you take away the 5' UTR from the virulent strain and put in the attenuated one, the virulence maybe drops a little bit. These results seem consistent and are statistically significant, but we need to do more work. But it does suggest there's probably at least a minor virulence determinant in the 5' UTR in terms of paralytogenesis. Next slide. We've also started to test ones. I showed you the right side didn't make a difference. What about the 2A or 2B or their combinations? And you can see putting in 2A, 2B, or both doesn't attenuate the virulent strain. And next slide. In the top there, you can see we're just starting to do the testing of the VP3 and VP1 proteins. And one of the cool results that we just got recently is the chimera that just has the VP3 from the attenuated strain really looks like it's attenuated. That should be 0% of the 10 mice. Sorry, it looks like it says 9 out of 10 there. And so we seem on preliminary data to think there's probably an important virulence determinant in VP3. We're also testing VP1 that has 3 changes and the combination of VP1 and VP3. I should also say that there's some very cool work going on from Raul Andino and colleagues doing the exact same type of thing, but a little different mouse strain that they're testing and mouse model, and a little different set of parental viruses. Next slide. We've also done some studies looking at how this virus spreads in neuronal cultures in cell chambers. And you can see here that we use human iPSC spinal motor neurons. You can grow them in these chambers, cross a grooved microwell center, and that enables you to isolate the cell bodies and processes and study infection in both compartments. Next slide. Virus happily grows if you infect in the distal compartment and spreads to the cell bodies. If you sever the axons or block axonal transport, you inhibit this. Next slide. It actually even spreads, and look at the green dots you see in those clusters of neurons from cell to cell through transsynaptic transport. The transport is predominantly retrograde, meaning back from the processes to the cell body, not the other way around. Next slide. Obviously, if you have a model like this, you can use it to test things like drugs. And so we've taken drugs like IVIG, which contains antibodies against Enterovirus D-68, and tested it for mouse protection. Now, sadly, I think a few lines are missing on this curve, but you can see if you use PBS in the white line, you get lots of disease and paralysis. And that pink line down there would be what would happen if you gave IVIG virtually at the same time as virus infection. And what you're not seeing there, is between the pink line and the white line, as you delay the timing of administration, you got less and less of an effect, but you could still see a therapeutic effect even 4 days or more after delaying IVIG from viral challenge. Next slide. This correlates wonderfully with decrease in viral titer, both in the site of inoculation, which is the muscle, and in reaching the spinal cord. Next slide. A group in China published survival data using mouse monoclonal antibodies against Enterovirus D-68. You can see the blue line at the top of the curve there is great survival with one of these antibodies called 15C5, and not so good survival in the other conditions. 15C5, as shown on the right-hand panel, sort of binds to a conformational epitope that involves the VP2 and VP3 proteins of the virus outer capsid. Next slide. If you stick that antibody into mice in our model, you can see that the red line compared to blue and green, that you dramatically improve paralysis results in treated mice. 15C5 was one of the other antibodies against outer capsid proteins that wasn't effective in that prior paper, really on survival, and HSV8 is a similar human mouse chimera. The human parts are all the constant domains, the mouse parts are all the variable domains, and using the same sequences as in that Zhang paper. And this is a collaboration with Zab Bio and Miles Brennan and his colleagues that I mentioned before. Next slide. We were very excited in testing that good antibody, 15C5, in a delayed model of paralysis. So here, this is more like the human real world, where you wait until mice start to show weakness using our paralysis score, and then begin to treat them intraperitoneally with antibody. And again, you can see the gap between the blue score, with the lower part being on the lower part of the graph, versus the black control. And so this is a great demonstration of what would really be a therapeutic model, and suggests, as does the IVIG story, that there's potential for immunotherapeutics and treatment, and of course, hopefully bodes well for vaccine design as well. I don't have time to talk about their work, but Matt Voigt at UNC has done terrific work with also humanized monoclonal antibodies with very, very similar results that they recently published. So there are other groups coming at it with a similar idea, again, with confirmatory findings. Next slide. We're also collaborating on antivirals, and I think I have 1 or 2 slides left, so I'll try to go through these expeditiously. There's a great group led by Jun Wang at the University of Arizona. They're biologic chemists and pharmacologists. They've been screening compounds for antiviral efficacy. Anna showed you a great slide summarizing some of the potential targets. They found that a hepatitis drug that had been FDA approved called Aptaviruses inhibited the viral 2A protease. And we've tested that in mice at doses similar to what the human doses were. And again, looking at those blue curves versus the orange and black ones on the left-hand side there, you can see that it was effective in treatment of mice. Now, probably telaprevir isn't going to be the right answer, but derivatives of it may be, and maybe even more effective. Next slide. This is just to make the point that one of the reasons I said that was the EC or concentration 50 is about 2 micromolar against the tough strain we tested it against. And although we were getting concentrations like that in our dosing model in muscle, we weren't really getting that good a concentration in the CNS or spinal cord. So higher doses and more penetrant drugs are probably the future. Next slide. I'll probably make this one the last one in view of the time, but I just wanted to show you that we've also worked with Jun Wang, who's very interested in targeting the viral 2C protein, which again, Anna mentioned in her introduction. And one of the best results that we've seen for an antiviral compound comes with one of these compounds that they've developed that targets the 2C protein called 6050-4, not a very catchy name. And you can see there how well that did in the blue line in terms of reducing paralysis compared to a control vehicle alone. And we're now in the process of testing that same compound in that therapeutic model. And also, of course, looking at its CNS penetration and pharmacology, which Jun is doing. So I'm sorry because of the Anna running the slides that I ran a little bit late, but I'm happy to stay on or answer questions, and just want to thank Anna and Richard for having me, and apologize again for my lack of brilliant WebEx technical skills. Thanks very much.
Thank you. That was fascinating.
Yeah, that was terrific, Ken. Thanks. I guess we can open it up for questions, and maybe I'll start, Ken. So, we didn't have an AFM, our big AFM spike in 2020, probably because of all the kind of social distancing mitigation strategies for coronavirus. But now that things are opening up, what's your prediction about this summer?
Yeah, I think I never honestly understood all the drivers of the biannual peak. I've seen great papers, which I don't fully understand the mathematical models of, I guess I'm not smart enough, that say you see these kind of patterns with other enteroviruses and attribute it to fluctuations in immunity in the susceptible group. I think if you had asked me, I think what you said is true, that it was our masking and social distancing and homeschooling and no daycare and things like that caused the gap in 2020. And if those models are right, all you've done is built up an even bigger susceptible pool. And so when you open the floodgates, you might predict it will come back big. Now, of course, we may have a little bit of a hybrid season because everybody's still grappling with, wait a minute, can we get rid of our masks? What are we going to do? So I think it'll be really interesting to kind of see how this plays out nationally. Like, is there a relationship between, areas which are more open and more like they were before and cases? But I think most of us would have predicted you will see this spike. It isn't that the virus disappeared. By the way, we saw the same thing with flu. I don't think we had hardly any flu cases in Colorado this year, and almost no admissions to Children's Hospital with flu. And the mortality in childhood flu dropped like a stone. Instead of hundreds of deaths in kids, there were virtually none. So it wasn't just an AFM thing. This is something we now know can block the spread of respiratory viruses and doesn't care whether you're an enterovirus, a flu virus, or something else.
Okay, so we had another question from Alexander Hilger. Could you please give some additional information or background on your mouse model?
Some additional information on the mouse model, Anna?
Yes.
Basically, we've used both outbred mice, NIH Swiss, and a variety of inbred mice, think C57 black and others, a variety of routes of inoculation. In our model, we deliberately tried to pick mouse models where you got neonatal disease, so that where baby mice developed a clinical illness that looked like the people. It's hard to get disease in adult mice, but if you do want to do it, the way you do it in most investigators' hands is by knocking out innate immunity in general and interferon signaling in particular. So basically, you take a little tiny 1 or 2 day old mouse, you stick a needle into the brain, the muscle, or an Eppendorf pipette into the nose. You inoculate them with the virus. You administer drugs or test things in baby mice by intraperitoneal inoculation. That sort of is the parallel in a baby mice to intravenous in a person. And then we just follow them blinded, doing things like a validated paralysis scoring system, or of course you take out, target organs and titer them for virus or prepare them for immunohistochemistry or do things like that. It's been published both in PLOS Pathogens and the Journal of Infectious Disease would be 2 good sources. And Richard and his group and us, I think David Brown was the lead author, if I'm remembering right, had a paper in MBio that also describes briefly some of the mouse experiments. So any of those would be good sources for more detail about the model.
Okay, and there was another question from Peter. Do you expect the viruses from 2016 and 2018 outbreaks to cause paralysis in your mouse models?
What a great question, Peter. We have been collaborating with a group at Wadsworth in New York who've been collecting isolates from 2016 and 2018. And we have started to test some of the 2016 isolates and have 2 of them for sure. They happen to be the first 2 we tested, so I can't give you a denominator that produced paralysis in mice. We've also tested 2018, 2 of the strains from Wadsworth, and I think the preliminary results are that neither of the 2 that we tested produced significant paralysis. So the preliminary answer is 16 for sure, some of the strains can, and I don't have a 2018 one yet that does, but we just have started testing some of them. And that was basically probably to answer Peter's question, that almost all the studies and strains that people have used experimentally are still part of the big 2014, cluster. Those are the ones that are best characterized and sequenced and so on. We have an ulterior motive for testing 16 and 18 because, of course, a cool way of validating stuff you did with site-directed mutagenesis or chimeras would be if you could find sequence correlates that matched what you had engineered among natural isolates, where the same change you said, dropped virulence or enhanced virulence, you could show in natural isolates that the same thing happened. So I think that's why we started to, let's, start to screen a few of those and see what happens.
Hey, Ken, the 2018 isolates, are they from AFM patients or just from respiratory disease?
No, they are just respiratory ones. It's sort of been hard to get isolates that are linked absolutely to AFM cases. To my knowledge, and I apologize if I'm misspeaking, but to the best of my knowledge, I think they were just clinical isolates, not AFM isolates.
Mm-hmm.
Okay, and then we have one more question. Was there year-to-year sequence variation in viruses from the same geographic location?
Ask me one more time, Anna.
Was there year-to-year sequence variation in viruses from the same geographic location?
So I think you guys, Richard, and you want to take that one. I'm going to say,
Sure.
what's the show where you're allowed to get a lifeline or something like that, or call a friend? I think the correct answer is yes. But what do you guys want to say?
Yeah, definitely. I mean, even within the same year, you can see different subclades kind of co-circulating in the same geographic region. So it's amazing how diverse this group of viruses is and how widespread it is. And, and even within a location, the diversity is pretty impressive. So yes, there is a lot of sequence variation from year to year, even within the same geographic region.
And I think it's been an interesting issue. There's a very good group, Vince Racconello and Amy Rosenfeld at Columbia, where there's been a little bit of back and forth in the field about whether the virus has changed and become more neurotropic. Why are we seeing this in the modern era and hadn't seen it really with the original strains? And do they use different receptors? Do they have different capacity to infect different types of neuronal cells? And I think that field, to be honest, is still evolving. I think Richard and I thought originally the answer was probably yes, that there were some things that were different about receptor utilization by modern and neurotropic strains. And Amy and Vince think the correct answer is no, that it isn't that the virus suddenly has changed its receptor ability to infect neurons. So I think the jury may still be out on that one. It'll be fun to see, what do we find? Can we identify paralytic determinants in vivo? And then, of course, what the hell does that mean? Does that mean that they can't, they differ in ability to kill or infect neurons? So, this may be a sort of backdoor approach to understanding that a little bit too.
All right, so if there are no more questions, then I think we could wrap up.
Thank you so much. It was really fun, and fun seeing all the people I hope we get to see live at some point.
Yeah, thanks so much, Ken. That was